Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Sudden oak death disease explained. Symptoms, causes, and management. Phytophthora ramorum Fungus. Table of Contents. Introduction. Causal Organism. Characteristics of Causal Organism. Symptoms. Disease cycle. Disease management. Introduction to sudden oak death. Sudden oak death is a plant disease caused by an oomycete pathogen that infects a wide variety of trees and shrubs, particularly in North America and Europe. The disease was first identified during the 1990s in California. It has led to significant mortality among oak and tanoak trees, resulting in serious ecological and economic consequences. The disease is primarily found in coastal forest regions of California and Oregon, USA. However, the pathogen has also been detected in several European nurseries and natural forests. Sudden oak death affects a broad host range of more than 100 different plant species. It is especially devastating to certain oak species, including tanoak, Notholithocarpus densiflorus, coast live oak, Quercus agrifolia, California black oak, Quercus kalagi, and shreve oak, Quercus parvula. Secondary host plants include bay laurel, Umbellularia californica, rhododendrons, camellias, and viburnums. While these secondary hosts typically do not die from the infection, they act as reservoirs that help the pathogen persist and spread in the environment. Causal organism of sudden oak death. The disease. Sudden oak death is caused by the pathogen Phytophthora ramorum, an oomycete, water mold, belonging to the genus Phytophthora. Although P. ramorum resembles fungi in its appearance and growth habits, it is not a true fungus. Scientifically, it belongs to the kingdom Stramenopila, Chromista, making it more closely related to algae than to fungi. The systematic classification of Phytophthora ramorum is as follows: Kingdom, Chromista, Stramenopila. Phylum, Umicota, water molds. Class, Peronosporomycetes. Order, Peronosporales. Family, Peronospораceae. Genus, Phytophthora. Species, Phytophthora ramorum. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Characteristics of Phytophthora ramorum. Phytophthora ramorum is a hemibiotrophic pathogen, meaning it initially infects living tissues, biotrophic phase, and later kills them to continue feeding on the dead matter, necrotrophic phase. The pathogen can reproduce both sexually and asexually, but its sexual reproduction is rarely observed in natural environments. It typically does not produce thick-walled oospores under natural conditions, indicating that it reproduces mainly through asexual spores, such as sporangia and chlamydospores. Sporangia. These are airborne or water-dispersed spores produced on infected leaves and twigs. They are spread by rain splash, wind, and contaminated materials. In moist conditions, sporangia release motile zoospores, which can swim in water and infect new host plants. Zoospores. These are motile swimming spores that actively move toward plant tissues, entering through natural openings or wounds. They are short-lived but highly infectious, requiring water for movement and survival. Chlamydospores. These are thick-walled resting spores that enable the pathogen to persist in soil, plant debris, or infected bark for extended periods, sometimes months or even years. They are crucial for long-term survival and reinfection of plants. Oospores. These are sexual spores that are rarely produced by Phytophthora ramorum. The absence of oospores in natural populations suggests that the pathogen relies primarily on asexual reproduction for propagation and spread. Download our microbiology study app made for researchers like you. Symptoms of sudden oak death disease. The symptoms of sudden oak death vary depending on the host species affected. In oak and tanoak trees, the most characteristic symptom is the appearance of bleeding cankers, which look like dark reddish-brown sap oozing from cracks or splits in the bark. Beneath the bark, the infected tissue turns dark brown to black, indicating extensive internal damage. Additional symptoms include browning of leaves, dieback of branches, and eventually the rapid wilting and sudden death of the tree. The disease is termed sudden oak death because trees often appear healthy until they die abruptly once visible symptoms manifest. In secondary hosts, such as bay laurel and rhododendron, the infection is usually latent or mild. These plants may show leaf spots, twig dieback, and minor branch damage, but they typically do not die from the infection. Despite showing limited symptoms, secondary hosts serve as crucial reservoirs for the pathogen, helping in its survival and spread to more susceptible tree species. Disease cycle of sudden oak death. Overwintering. Phytophthora ramorum survives in the environment for long periods by producing chlamydospores, which are thick-walled resting spores. These spores allow the pathogen to persist in soil and plant debris, even under unfavorable environmental conditions. The pathogen can also survive asymptomatically in hosts such as bay laurel and rhododendron, which act as reservoirs carrying the infection without being killed by it. These asymptomatic hosts enable the pathogen to persist and spread silently in the ecosystem. Dispersal and spread. The main means of dispersal for Phytophthora ramorum is through sporangia, asexual spores formed on infected leaves and twigs. These spores are spread by rain-blown winds, allowing both short and long-distance transmission. Runoff water contributes to soil contamination and root infections in nearby plants. Human activities such as transporting infected nursery plants, contaminated soil, or firewood play a major role in spreading the pathogen to new areas. Mechanical transmission may also occur when insects or animals carry spores on their body surfaces. Host penetration and infection. Upon reaching a susceptible plant, the pathogen enters through natural openings like stomata, lenticels, and hydothodes, or through wounds caused by pruning, insect feeding, or environmental damage. In highly susceptible species, such as tanoak and oak, the pathogen can directly penetrate the bark. Once inside, zoospores incist and germinate, forming germ tubes that establish infection within plant tissues. Colonization and disease. Development. The pathogen spreads internally through the vascular tissues, bark, and cambium interrupting water and nutrient transport. It secretes enzymes and toxins that kill plant cells, leading to necrosis and tissue damage. In oaks, bleeding cankers develop, appearing as dark reddish-brown sap oozing from bark cracks. Internally, the vascular tissue becomes brown to black, indicating severe infection. On secondary hosts like bay laurel and rhododendron, necrotic lesions and twig dieback are commonly observed. Sporulation and secondary infection. Once established, the pathogen produces new sporangia on infected plant surfaces. Under moist conditions, these sporangia release zoospores, which initiate new infections. In dry conditions, they may remain dormant until the environment becomes favorable again. The secondary spread occurs when sporangia are dispersed by rain splash and wind, infecting nearby plants. Asymptomatic hosts often play a key role in secondary infection cycles, producing spores without showing severe symptoms. Late-stage infection and tree death. In highly susceptible trees, such as tanoak and coast live oak, infection progresses rapidly, causing vascular blockage and decline of the tree. As trees weaken, secondary invaders like bark beetles and opportunistic fungi, e.g., Hypoxylon thorsianum, accelerate death. Within months to a few years, infected trees wilt, brown, and die, becoming part of the inoculum source for further outbreaks. Dormancy. Under unfavorable conditions, such as dry or hot weather, Phytophthora ramorum produces chlamydospores and enters a dormant stage. These durable spores allow the pathogen to survive in soil, fallen leaves, and bark debris for extended periods. When environmental conditions become favorable again, the pathogen reactivates and resumes its life cycle, initiating new infections. This long-term persistence in plant debris and soil makes the disease extremely difficult to manage and eradicate. Sudden oak death disease management. Controlling sudden oak death is challenging due to the rapid transmission of Phytophthora ramorum through rain, wind, water, and human activities. Since there is no cure for infected trees, management focuses on prevention, early detection, containment, and mitigation to reduce further spread and damage. Quarantine and regulatory measures. Quarantine regulations are one of the most effective strategies to prevent the spread of Phytophthora ramorum. Governments have restricted the movement of infected plant materials, soil, and nursery stock to stop long-distance transmission. The United States Department of Agriculture and the European Union have established quarantine zones enforcing strict regulations on plant transport from infected areas. Regular surveys of nurseries and forests help in early detection and rapid intervention to limit outbreaks. Sanitation. Since Phytophthora ramorum can survive in soil, water, and plant debris, strict sanitation practices are vital. Removing and destroying infected plant material minimizes sources of inoculum and reduces disease spread. In forests and landscapes, pruning diseased branches and removing infected trees helps slow the outbreak. Disinfecting tools, machinery, and footwear before moving between sites prevents accidental spread. In nurseries, containers, irrigation systems, and soil should be regularly cleaned and sterilized to reduce contamination risks. Chemical control. No chemical treatment can cure infected trees, but some can protect healthy ones and slow disease progression. Phosphite, phosphonate, Treatments strengthen tree defenses and are typically applied as trunk injections or bark sprays in high-risk areas. Fungicides such as mefenoxam and metalaxyl may be effective in nurseries, but are ineffective for large forest trees. Copper-based sprays can help suppress spore formation on infected ornamental leaves to some extent. Biological control. Still in the experimental phase, biological control shows promising potential. Beneficial microorganisms, such as Trichoderma species and certain soil bacteria, may suppress pathogen growth in the environment. Research into developing resistant tree varieties through selective breeding or genetic modification is ongoing as a long-term management solution. Cultural controls. Removing highly susceptible tree species from infected sites prevents them from serving as reservoirs. Thinning dense forests enhances air circulation, reducing moisture and slowing disease development. Planting less susceptible species around infection zones creates buffer areas that limit pathogen spread. Avoiding unnecessary wounding of trees during activities such as logging or landscaping reduces potential entry points for the pathogen. Resistant plant species. Certain tree species show natural resistance or tolerance to Phytophthora ramorum. Some oak species and bay laurels possess partial resistance, making them valuable for restoration and replanting efforts. Monitoring and early detection. Regular monitoring helps in identifying infections early before widespread damage occurs. Aerial and ground surveys are conducted to locate trees showing bleeding cankers, leaf blight, or dieback symptoms. Prompt detection and response can prevent the disease from spreading to new areas. Public awareness and education. Educating landowners, gardeners, foresters, and the general public is crucial for disease control. Awareness programs inform people about symptoms, quarantine procedures, and sanitation practices. The public is strongly advised not to transport firewood, soil, or plant debris from infected or quarantined zones. Community participation enhances control efforts, as informed individuals can actively protect their local environments. Integrated disease management. An integrated approach combining regulatory, cultural, biological, chemical, and sanitary practices is the most effective strategy for managing sudden oak death. Using multiple control methods simultaneously helps reduce pathogen spread and mitigate damage more efficiently than relying on any single technique. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!